Hello Interstellar to all the class 12 students. So this is going to be the final video of our chapter number 2, sexual reproduction in flowering plants. Right. And in this particular video we are going to discuss about post-fertilization events. So what happens after fertilization in flowering plants? And remember, in flowering plants, fertilization is also referred to as double fertilization because there is involvement of two fusions, syngamy and triple fusion. Therefore, fertilization in flowering plants is also called as double fertilization. So we will see what happens after double fertilization in flowering plants. Right. In post-fertilization events, we have to look at the development of these structures like embryo, endosperm, seed, and fruit. Now, this is pretty important. Embryo, it develops from zygote. So zygote, it has formed from the fusion of one of the male gametes with the egg cell, which is called as syngamy. Syngamy results in the formation of zygote, and zygote, it will develop into embryo during post-fertilization event. And then we have got endosperm, which has developed from primary endosperm cell, and primary endosperm cell is the result of triple fusion. So one of the male gamete, it fuses with the polar nuclei of the central cell, and that is called as triple fusion. And primary endosperm cell, which is formed during triple fusion, it develops into endosperm during post-fertilization event. And then we have got seed, which develops from mature ovule after fertilization. And we have got fruit, which develops from ovary after fertilization. So the formation of all these structures, right, after fertilization are the part of post-fertilization events in flowering plants. Now first let us look at the formation of endosperm. So endosperm develops before embryo. This is very important. Right? Endosperm la ishtoa. The embryo gi nyela develop chagu gores. The kharchin re law imbaina. Endosperm gi lengen di kosyo gor. Endosperm gi lengen kharre. It provides nutrition for the developing embryo. So embryo la the develop chevala endosperm gor injimiji. So thangbu embryo develop chigre. Amena endosperm develop chigre lawa imbana thangbu nyala khar develop chegu gore endosperm develop chegu gore. Clear chungswa. So endosperm development precedes the development of embryo because a certain amount of endosperm has to be formed for the development of embryo to begin, because endosperm provides nutrition for the developing embryo. And endosperm khandis form chigre lawa imbana. The primary endosperm cell becomes the triploid endosperm tissue. So endosperm tissue lash mindwe ti khane form chigor lawin bena primary endosperm cell ne form chigor. So the primary endosperm cell, it divides to give rise to a tissue called endosperm tissue, and the cells of this particular tissue is triploid. Triploid means it contains 3 sets of chromosomes. Nucleus sum fuse chigi wa sonza and endosperm nalo yogi cell dige sama kharre triploid re. Now, the cells of this tissue is filled with reserve food materials. I have told you already, right? So the function of endosperm is to provide nutrition for the developing embryo. So therefore, the cells of this particular tissue, endosperm tissue, is filled with reserve food materials. Now, there is one interesting fact over here, right? One important information, and interesting information. You all must have consumed coconuts, right? Fresh, tender coconuts. You must have drank coconut water. You must have eaten this white kernel as well. Both of these, they are endosperms of the coconut plant. Coconut plant ge endosperm re da dingi ga. Chu de endosperm re. Kang saya kaush to de khare endosperm re. Ta dingi para la khewa khare yore la wim na. The coconut water is free nuclear endosperm, whereas the one which you eat, the kernel, the white kernel, it is the cellular endosperm. Free nuclear endosperm means when the nucleus, it divides freely without formation of cell wall, it is called as free nuclear endosperm. So when the nucleus of primary endosperm cell, it divides repeatedly without formation of cell wall, that is called as that results in the formation of free nuclear endosperm. And when the cell wall forms, it becomes cellular and it becomes the cellular endosperm, which is the white kernel. Okay, so this is endosperm ki lengen khar re to embryo develop chewe kabla khola nutrition te re do. Okay, the next is embryo. Embryo, it develops from zygote. So the zygote, which results from syngamy, it develops into embryo at the micropylar end of the ovule. Zygote the kharcha dure, embryo la gyurdur. So zygote the embryo la gyurdanj minde. The kiranzu remudinal teorta. You can find this particular diagram in your textbook. So in your textbook, you can see that the two synergids, they will degenerate. At the micropylar region, the two synergids of the egg apparatus, they will degenerate. The egg cell which has fused with one of the gamete, male gamete, the zygote will develop into embryo. Okay, and this primary endosperm nucleus, it will develop into endosperm, whereas the antipodals, they will also degenerate. The shuma ne kira zu thiwa ta wi na synergids ke degenerate cha dure, antipodals degenerate cha dure. Post-fertilization alone tewa yo khare la wi na zygote the embryo la yu dure ani tela primary endosperm cell jorwa ti khar la yu dure endosperm la yu dur tissue la. Now let us look at the zygote over here, which is diploid. The zygote, it will gradually develop into an embryo. Embryo nyam develop chedan di del tyon yorda. Tangbo kharji do cell chik di cell ni le gyurgu do. Tene cell mangbo le gyurgu do ni ne shi ne gelan cha du ro. So it becomes a globular embryo, and then it becomes a heart-shaped embryo, and then it becomes a mature embryo. Okay, so embryo la di khane chungyor cell tagarane chungyor da zygote la cell dine chungyor. Okay, so from a diploid zygote, it becomes a multicellular embryo. Now let us look at the structure of embryo of the flowering plants. In flowering plants, we have got dicot and monocot. So we have got two different types of flowering plants, dicotyledons and monocotyledons. So how do they get their name? If you see over here, in the plant, in the dicot plants, the embryo of the dicot plant looks something like this one over here. Right? It has got two cotyledons. Cotyledon near. Monocot lana khala ore cotyledon chikma do yomar. So we will see what it means. If you look at the structure of a dicot embryo, you can see that it has got two cotyledons, and cotyledons are nothing but embryonic leaves. Right? Embryo now lomado sotanyi jiyore. It helps in the provision of nutrition to the developing embryo. So these cotyledons, they are like embryonic leaves. Embryo ki lomado chadu. And cotyledon ni yoba sonza. Sishin dige rilanga zu khalla ore dicotyledons la or. So cotyledons ni yoba sonza dicot la or. Now if you look at the entire structure of the embryo, you have got an axis. Right? Let me write over here. You have got an entire axis over here. The axis in bachasha. Embryonal axis reda. Okay, so cotyledon ni de do. Cotyledon chik de che, cotyledon chik de do. Now the one which is above the cotyledon, right? The one which is above the cotyledon, that is called as epicotyl. Epicotyl le zambalo cotyledon gi gang le yo. And epicotyl gi top de la khari ore la wi mena plumule la dir. Plumule is nothing but the stem tip. Later on, when the embryo develops further, right, this particular portion will become the stem. Stem theongure. So cotyledon ki level ki gang la yo dila epicotyl che. Epicotyl ki top de la khari ore plumule yore sro. Plumule di khar la gyurdu ore shuma, it will become stem. And cotyledon ki level ki wall yo. The portion of the embryonal axis below the level of cotyledon, it is called as hypocotyl. Right, below the level of cotyledons. And hypocotyl ki wa de la. Hypocotyl ki wa de la khari ore we have got radicle, which is nothing but the root tip. This will develop into roots later on when the embryo becomes mature or seedling. Right. So radicle che. And the radicle kitse de la khari there is a root cap. So since it has to go down and penetrate into the earth, it has got a protective cover, which is nothing but the root cap. Radicle the root chadure, root kitse la khari there is a protective sheath called root cap. So this is the structure of dicot embryo. Dicot embryo yakushin jangu word structure. And if you look at the monocot embryo lata we mana monocotyledons, grasses, maize, sugarcane, dige sama khare monocots re. So monocotyledons, they have got only one cotyledon, and in grass family, the cotyledon is called as scutellum. The cotyledon reda. the embryonic leaf dire. And here we have got the embryonic axis. So embryonic axis la parchi khardo shoot apex slash do, which is above the level of cotyledon. Shoot apex che, shoot apex ki gang la khardo coleoptile, which is nothing but it has got enclosed leaf primordia. So leaf primordia yo de ngazo khala ore coleoptile la ore. Okay, and below we have got the radicle, the same root tip, the root tip, and then we have got the root cap, and root cap ki gang la there is a protective sheath called as coleorhiza. So these are the structures present in the monocot embryo. So you have to know the two structures, right? The structure of dicot embryo and the structure of monocot embryo. Now, after the formation of embryo, right, we have to now look at the formation of seed. So seed, after fertilization, ovule develops into seed. So ovule structure. So ovule layer after fertilization seed it will develop into seed. Now there are two types of seed, albuminous seed and non-albuminous seed. Kisi seed nanglo la, seed nanglo la endosperm la deyo na. After embryonic development, endosperm la deyo na. Then they seed rig lang albuminous seed lagre. For example, wheat, maize, barley, castor, all these seeds, they are called as albuminous seed because there is some leftover endosperm in it. Whereas non-albuminous seeds are those seeds which does not contain any endosperm remnants. Right? So, example kare pita, groundnut. So these seeds, they do not have any remaining endosperm in it. So these are the two types of seeds. Now, seed nanglola, you must have heard about the tough seed coats. Seed ki coat jo marwe. Ti khane chungyo re law imba na. The integuments of the ovule, they give rise to seed coat, and seed coat protects the embryo. Right. Now, seed coat the complete marda. There is a small pore. Small pore is nothing but micropyle. Micropyle, it remains as a pore on the seed coat, right? And micropyle ki it is for the entry of oxygen and water into the seed. Right? Since there is a developing embryo inside, the developing embryo requires oxygen as well and water. Right? Ko develop cha wala oxygen the water go gore, and oxygen the water the seed coat ki gune zutu marwa. So seed coat gana igun yo yecho mare micropylar region dela. So igun ne ya zutu gore. And if the conditions are favorable, the seed, right, it will germinate. Embryo develop cha and seed germinate chen khachire sishin thoyong gorwa. But if the conditions are not favorable, the embryo inside the seed, it may enter into a state of dormancy or inactivity. Embryo dikhachire ko lega inactive chadur. Inactive chana and seed dikhar chare dormant chadur. So the seed, it waits for favorable conditions. Seed dikhar chi gore favorable condition go de yor hote vor. And each hawa jab temperature sama takta yo bhi na, the seed will germinate later on. Is it clear? So seed che. So we have these structures of the seed, right? In the textbook, you are given with the structure of the seed over here, right? Pea plant ka jigi seed ta, ani maize ka jigi seed ta. Tige sama kira zulu te yorta. So you have to study the structures of the seeds over here, right? Endosperm lana khare. It provides nutrition for the developing embryo. Scutellum lana monocot ki khare. It is a cotyledon of the monocot, like grass family. It is called as scutellum. And we have got the plumule, which becomes stem later on. We have got radicle, which becomes roots later on. Stem ki gang la khari coleoptile yore. Root ki tip gang la khari coleorhiza yor. Okay, so structure dige sama kena ziyabu kocho vechiya. Now, what are the advantages of producing seed? Angiosperm ki seed produce chene advantage khar khari yore lawo yimba na. Seed dispersal latch the seed, they get dispersed, right, far away in different areas. The seed will get dispersed. Some seeds, they are carried by animals. Some seeds, they are carried by wind. Some seeds, they are carried by water. Therefore, the seeds, they are getting dispersed. And when the seeds gets dispersed, the plant species, they get dispersed. And it gives advantage to the plant. the plants, they can now occupy different areas. Right? So that is dispersal of the plant species, and it is helped by seed dispersal. So that is one advantage. 사실 like there we na resource la competition shuk chambu yung gro saja manda la kya ba na ani plant gi survival la kyapsa yora. So dispersal of plant species is very important, and that is provided by seed dispersal. Advantage chikche advantage niwa dikhare la wi na seed nanglo la khari ore endosperm da dige sama yoro. So it provides food reserve for the young seedling di khora photosynthesis che gi potential male pado capacity di male pado ra kharchi gore seed ki kola food reserve te or so it is giving a head start to the young seedling. It provides food reserve for the young seedling so that the plant can survive. Right, and the seed coat, it protects the young developing embryo. So embryo develop cheve kabla seed coat ki kharchi gore protection te or di sumba diche. Right, and inside seeds the embryo can remain dormant. If the outside conditions are not favorable, the seeds will remain dormant. But inside the embryo, even though it is inactive, it remains viable. It is not dead. It remains viable. So seed viability is very important. Some of the seeds, they can stay viable for a very long periods of time. And the kechumbu be chadu gorda. Agriculture shedu be kechumbu chadu gor. Kachin nana azu se nya gora. Next season chedu nya gor. Lo ni che chedu nya gor. Dinde de zambala kesi seed la dormant we cannot store seed. If we cannot store seed, right, agriculture chetu marwa. So the advantage chikchaswa. And example neater. Some seeds, they can remain viable for a very, very long period of time. For example, Lupinus arcticus slash midwe. Koigi seed dida lo tongda chugijela. It is still alive. It is still viable. This is sendi. Tongda chugijela mata bimba ko hachi or keg nubayor. And then there is another plant, Phoenix dactylifera lash. Koigi sendi lo tongda nigijela nier. Estimated lo tongda nigijela niere. Sendi tabnet handu hachi or zishin ti kegor. So these seeds, they can remain dormant and viable for a very long period of time. And it's a very important character. So these are the advantages of the seed. Okay. The next kare, fruit, the development of fruit. Fruit fruit, it develops from ovary. The mature ovary, it will develop into fruit after fertilization. Right. Now, the walls of the ovary ki walls they will develop into pericarp. Pericarp is nothing but the wall of the fruit. Right. Am ki pakpato di kharchadu pericarp to chadur. Chilogi pakpato minde shingdogi pakpasto the pericarps. Right. It develops from the walls of the ovary. Now the fruits, they can either be fleshy like in mango and guava, or it can be dry, for example, groundnut. Now, very important, from your examination point of view, fruit is of two types, true fruits and false fruits. True fruits are those fruits which develop only from the mature ovary. Shingdo ovary chekpo ne chagyo yimba na tilang azu khalere true fruit lagre. Whereas false fruit like apple and strawberry, the thalamus of the flower contributes in the formation of fruit. Shingdo chawala ovary chekbo mayimba che ani medo ki thalamus la jor ta chela kiranzu medo khaji structure ta da medo khaji ki thalamus ki ani fruit formation la help chigi yobi yimba na. And the fruit it is called as false fruit, whereas true fruits, it develops only and only from mature ovary. So false fruit ke example te lawi by na apple da strawberry tena digre cashew geyor. Okay, so that is about one fruit. Now parthenocarpic fruit ledger. So these fruits, they develop from fertilization, right? After fertilization, right? When the ovary develops into fruit after fertilization, so the types the true fruits, the false fruit. Now, there parthenocarpic fruit lash. Parthenocarpic fruits are those fruits which develops without fertilization. Fertilization machung ne ani kesi o khasa fruit chungu yobi ba na ani fruit denzu rigla ngasu khalagre parthenocarpic fruit lagre. right? And parthenocarpy can be induced through application of growth hormones. Growth hormones, plant growth hormones shukchambu tewa by na. Any plants without fertilization, they will start producing fruits, and that is called as parthenocarpy. The tembaya bunya parthenogenesis the daudo cha do. And the fruits, such fruits which uh, forms from parthenocarpy, such fruits are seedless. You must have heard about seedless oranges, seedless grapes, right? And banana is a good example of parthenocarpic fruit. It does not have any seeds inside. Okay, so fruits which arises without fertilization, which develop without fertilization, it is called as parthenocarpic fruits. The final topic in the law. Video this final topic dikhare apomixis. Apomixis lana khala gore. The mechanism of the plant to produce seeds without fertilization is called as apomixis. Okay, that di the matua. Parthenocarpic fruit da apomixis matua. Parthenocarpic fruit lana khala gore. Parthenocarpic fruit la disambala. The development of fruit without fertilization is called as parthenocarpy, whereas apomixis is the development of seed without fertilization. Production of seed without fertilization is called as apomixis. In seed yomarta. Development of fruit raw. Kisikela dimatsu wimenekela mare. Whereas here, right, some plants they produce seeds without fertilization, and seeds can develop into a new plant. Right. So apomixis here. So apomixis is a form of asexual reproduction, which mimics sexual reproduction. Sexual reproduction ki lendo che yoro mimicry lana khalo lendo che wala goro. So sexual reproduction lendo che ngedo chadu goro apomixis. It is a asexual reproduction that mimics sexual reproduction. Very important concept. Okay. So apomictic seed contains produce chigre in plants ki. Right. Some plant species, the egg cells they are diploid in nature. Namju egg cell khari haploid ra. In this particular case, right, some plants they produce diploid egg, and that diploid egg it develops into embryo without fertilization and gives rise to seed. Di thaplam chigre. Thaplam niwa khare lawim in some plant species, right? The nuclear cells, nuclear cells, the ones which provide nutrition, right, for the developing embryo sac. The nuclear cells, some of the nuclear cells, they will develop into embryos, and nuclear cells they are diploid in nature. Nuclear cell dige kisi embryo chadu yo bim na yan dia khare apomixis re. And this will give rise to seeds. And khonzo la genetic variation layer chance koro yo marwa because it has formed without fertilization. And in the second case, kisi nuclear cell mangbochi ani embryo lo gyuwa change chagu yo bim na. Then what will happen? There is a formation of polyembryony. Polyembryony lana khala gore occurrence of more than one embryo in a seed. Kisi suen chig na lola embryo chigle manga yobi na dila khale polyembryony le. It can happen when the nuclear cells develops into embryos. Nuclear cells dige sama embryos lo gyurwa imba na. So nganzu ki seed form che imba na, and seed na lola embryo khachire embryo mangbupe form chire. Okay, so that is polyembryony. Now I have got one assignment. Okay, so you just read the final paragraph of your chapter and write down a short note on the advantages of apomixis. Apomixis slash the production of seeds without fertilization. Apomixis the any kharchin advantages res, especially for farmers. Anzugi rural farmers chedula apomixis the kharchin kechumburido. Advantage kharite do. Di kana note chinju so read the final paragraph of the chapter and prepare a short note on the advantages of apomixis for Indian rural farmers. And with this, we have come to the end of the chapter. Right. From next video onwards, we are going to discuss about chapter number 3, human reproduction. Right. And along with that, you have to also complete the exercise questions provided in your textbook. Right. The textbook now your exercise question they get some complete chida. So next we will start with human reproduction. Okay, thank you.